So moving to example number 2 of dihybrid cross. Okay. So dihybrid crossing. Crossing, I told you, breeding of two different plants having two contrasting characters. Contrasting means opposite character. So here I have written green inflated crossed with yellow constricted. So what are these green and yellow? Green and yellow is actually the pod color. Okay, pod means this is pod. Inside this seeds will be present. Okay, the covering. That is what is called as the pod. So in one plant, the plant is having a green colored pod. The other plant is having a yellow colored pod. Whereas the second character that we are considering in the same plant is inflated and constricted in the other plant. So what is this inflated and constricted? Inflated and constricted is actually the shape of the pod. Inflated means the pod will be having the color like this. Constricted means the pod will be having the shape like this. Okay? So that is the meaning of inflated and constricted. So in this, how many characters do we consider? We are considering two different contrasting characters. One is the color of the pod, another is the shape of the pod. Okay, already I have written pod color and pod shape. So green, how will you represent green? Capital G, capital G. Inflated, capital I, capital I. Excuse me. Now yellow, small g, small g. Constricted, small I, small I. Okay. Now coming to the gamete. What is the gamete produced in the first plant? Capital G and capital I. From both this capital G, 1 G. From both the capital I, 1 I. So that is the gamete produced in the first plant. Now what are the gamete, or what is the gamete of the second plant? This is the gamete. From both the small g's, 1 G, and from both the small i's, 1 I. Okay, so these are the gametes. Now after gamete formation, what will happen? This will combine together so as to produce the F1 generation, or first filial generation. So I told you, first you concentrate on the G's. Okay, first we will write capital G, then small g of the next plant. Then come to the I. Capital I of this plant and small I. Okay. Now going to the next is the phenotype and genotype. What are the phenotype and genotype? Phenotype, I told you, it is the appearance of the plant. Here capital G and capital I is present. If capital letters are there, what will be the appearance? Concentrate on this capital letters character. What is this capital letters character? Green inflated. Okay. So what is the phenotype? Phenotype is green inflated. Now what is the genotype? Genotype, you copy this like this, or you can write heterozygous, green and inflated. Okay, this is known as heterozygous condition. Heterozygous means the genes are different for a particular character. Now moving to F2 generation. What you have to do in F2? Crossing this. Capital G, small g, capital I, small I, crossed with capital G, small g, capital I, small I. Okay, so what are the gametes produced? First we will concentrate on the capital G. Capital G will combine with capital I. This is one gamete. So I am writing here gamete. Capital G will combine with the small I, that is the next gamete. Next is small g combined with capital I, third gamete. And small g combined with the small I, that is the fourth gamete. The same is the case in the second plant because the second plant's genetic makeup is also the same. Okay, so here again what are the gametes? Capital G combined with capital I, capital G combined with the small I, small g combined with capital I, and small g combined with small I. Okay, now what is the next? We are supposed to draw the Punnett square. So drawing the Punnett square, which comprises of 16 squares. Okay, so I am going to draw that 16 squares. This is the dihybrid cross. So 16 squares. Here I will write the male. Here this side I will write the female. So let this be the male plant. So capital G, capital I first. Next gamete is capital G, small I. Third gamete is small g, capital I. Fourth gamete is small g, small I. Okay. Now moving to the next female plant's gametes. Again capital G, capital I. Capital G, small I. Small g, capital I. And small g, small I. Don't change the order, okay? If you are writing this in this order, the next also you write in the same order. Now, just crossing. G, G, I, I. So write G, G, I, I. What is this color? Green, inflated. Okay, I am not writing the full. Now G, G. Capital I, small I. I hope it is clear to everyone, okay? Again green, capital I is there, so it is inflated. Next this and this. Capital G, small g, capital I, capital I. Even though small g is there, capital G is also present. So this is also green, inflated. Now this into this. So capital G, capital I, small I. Heterozygous condition, but here capital G and capital I is there, so it is again green, inflated. Okay, so first row is over. Moving to the second row, this crossed with this. So capital G, then I and I. Again green, inflated. Okay, capital G, small I. So, what is it? It is green, constricted, because small I, both are small. So, if no capitals are there, you concentrate on that, it is constricted. Okay? Now, this into this. So, capital G, capital I, small I. Again, green, inflated. Now, this into this. So capital G, small I again. So what is it? Green, both are small i's. So small, constricted. Moving to the third row. Okay, this into this. First you, even if this is the small letter, here capital letter is there. So first you write the capital letter. Okay, so capital G of this, small g, then capital I. So again green, inflated. Now this into this. So capital G, capital I, small I. Again green, inflated. Now this into this. So small g, capital I. Both are small g's. What does that small g stand for? It is yellow. So yellow, inflated. First we got. Now this into this. Again small g, capital I, small I. So again it is yellow, inflated. Okay. Now moving to the last row. This into this. So capital G, capital I, small I. Again green, inflated. Now this into this. So capital G, small I, small I. It is green constricted. Now moving to the last. Small g, capital I, small I. What is it? Small g is yellow inflated. And the last one is this. Small g, small I, small I. So you will see green inflated. How many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So right here, this is the phenotype. Green inflated 9 is to how many green constricted are there? So green constricted. Here 1, I am drawing a star for green constricted, 2 green constricted, again 1 more. So that is 3 green constricted is to how many yellow inflated? So yellow inflated. What we are writing? What we are writing here is the phenotype, okay, of F2 generation. So yellow inflated, I am drawing one circle. So here one yellow inflated is there, here one yellow inflated here. So how many? 1, 2, 3. So yellow inflated is 3 is to last one, it is yellow constricted. I am just drawing a line. So yellow constricted, how many? That is 1. So what is the ratio? 9 is to 3 is to 3 is to 1 is the phenotypic ratio of a dihybrid cross. This will be the same always. Okay. So phenotypic ratio of dihybrid cross will be always 9 is to 3 is to 3 is to 1. Now what about the genotypic ratio? The genotypic ratio is very complex. Now sometimes in the question paper, even without drawing the Punnett square, sometimes you will be asked to write the phenotypic ratio of F2 generation. In that case, how will you write? I am just rubbing all this. I hope the concept is clear to everyone. Okay, I will tell you an easy method for writing that phenotypic ratio. First thing is that you have to by heart this ratio, that is 9 is to 3 is to 3 is to 1. This you need to by heart. Because I told you it's after all 16 squares. So 9 plus 1 is 10, 10 plus 3 is 13, 13 plus 3 is 16. Okay, so this is making up the ratio which is equal to 16. Okay, 16 squares of the Punnett square. Now, you have to by heart this, but you have to write that phenotype, isn't it? So imagine that this is the same condition. So what you have to do is, the extreme ends take that numbers. The extreme end number is 1 is 9, the other one is 1. Okay. So 9 will be the first dominant character that is given in this crossing. So there will be 9 dominant character plants. So which is the dominant character here? Green inflated. So green inflated, how many plants will be there? There will be 9 plants. Then I told you about the extreme numbers first. Okay, first we are bothered only about this extreme numbers. So 9 is this dominant parent. Okay, green inflated. What about this 1? 1 will be the recessive parent's character. So this one will be this recessive parent, that is yellow constricted. Understood? So we got the 2 extreme ends. So if you are crossing 2 plants or if you are breeding 2 plants having 2 contrasting characters, 9 will be showing the dominant character and only 1 plant will be showing the recessive character. Now what about these 2 trees that we are written in between? So this is very simple. What you have to write is, I told you here we are having 2 characters. So for this first tree, take the first character. Which is the first character here? That is green. Combine green with the second character of the second plant. What is the second character of the second plant? Constricted. Okay. So 3 will be green constricted. What about the next 3? Here, which is the next character? So green and constricted is over. Okay, now what is remaining? Yellow inflated is remaining. So this 3 will be, take the second plant's first character, yellow, and the second character of the first plant. So yellow inflated. Okay, so even without writing the Punnett square, if you are asked to, even without drawing the Punnett square, if you are asked to write the phenotypic ratio of F2 generation for a dihybrid cross, you have to keep in mind it is 9:3:3:1. Then you have to write along what is that 9 belongs to. Okay, so simply write that dominant character for 9 dominant character will be there. 1 recessive character, then mix these 2 characters, 3, 3. That means first character of the first plant, mix it, not a mixing, first character of the first plant combined with the second character of the second plant. Similarly, first character of the second plant combined with the second character of the first plant, that will give you these 2 3s. I hope the dihybrid crossing is clear to everyone. Moving to the next example, I hope the previous example is clear to everyone. Now example number 3, heterozygous condition. Okay, we were discussing only about homozygous condition. Now heterozygous tall purple crossed with dwarf white. I am just showing one example, okay, for you to understand. So heterozygous tall purple, how will you represent the heterozygous condition? Tallness. Homozygous means it will be both capital T's. So heterozygous means one will be capital T, another will be small t. Purple, one will be capital P, another will be small p. Why we are writing the genes like this? It is because it is already written heterozygous. So hetero means different. So heterozygous conditions, you will be having the genetic makeup like this. Dwarf white, it is a recessive character, so there is no change. Dwarf is always small t, and white is this opposite letter, that is small p, small p. Okay, now moving to the gamete. Here 4 different genes are there. So first we will write capital T combination. Capital T will combine with the capital P. Capital T will combine with small p. Small t will combine with capital P. Small t will combine with small p. Okay, so 4 gametes are produced in the first plant. Here both are same, so only 1 T. Here both are same P, so only 1 P. Okay, now what you have to do? Here Punnett square we cannot draw. Gametes number is not the same. Okay, so what we have to do is simply combine this like this. First gamete combining with this. So TP you can write 4 times, no problem. Okay, same TP like that you can write 4 times. That is also the same. So instead I am showing like this. So capital T, then small t of the next plant, capital P of the first plant, small p of the second plant. I told you in order to avoid confusion you can write the same thing 4 times because here 4 are there. Okay. Now next is this and this again. So capital T of this plant, small t, then both are small p's. Now the third one and this. So both are small t's and 1 capital P and 1 small p. Now the last one is this. Small t, small t, small p, small p. Now what is the appearance of this? This will be heterozygous purple, that is phenotype. Okay, phenotype is, tall purple. Genotype you can write the same or you have to write heterozygous purple. What is the phenotype of this? Tall white. Why? Because both are small p's. So it is white. What about this? Both are small t's and there is one capital. So this will be dwarf purple. And what about this? Dwarf white. Okay. So phenotype means appearance. As capital T and capital P is there, it is tall purple. Here one capital T is there but both are small p's. So it is tall and white because small p is for white. Here both are small t's. Small t is for dwarfiness. One capital P is there. So if capital is there, that is standing for purple. Here both are small t's and both are small p's. So it is actually dwarf white. Okay, this type of crossing will not come for the exam. This is just an extra idea that I am providing to you. Okay, how you will be doing a heterozygous dihybrid crossing. So we will not proceed into the second generation now. Okay, within this first generation itself I am stopping. So this is heterozygous condition. I hope the monohybrid crossing and the dihybrid crossing is clear to everyone. Monohybrid crossing, the phenotypic and genotypic ratio is important. Dihybrid crossing, phenotypic ratio is important. Genotypic ratio is actually not mentioned in the syllabus. So phenotypic ratio of a dihybrid cross, I told you many times, it is 9:3:3:1. So you have to remember all this monohybrid phenotypic and genotypic ratios and dihybrid phenotypic ratio. Genotypic ratio, if dihybrid cross, if it is it will not come because it is not there in the syllabus, but for your extra understanding, genotypic ratio is actually very complex. Okay, that is the answer for genotypic ratio of a dihybrid cross. I hope the concept of crossing is clear to every one of you.